Hello, my name is Dr. Luke Bell. I'm lecturer in molecular horticulture at the University of Reading in the United Kingdom. This is a screencast of material originally made available for the module Principles of Horticulture and Crop Production from the years 2020 to 2025. Please note that some of the details may now be outdated.
So during this screencast you're going to learn or understand the basic life cycle of horticultural crops, describe the different states of seed viability, describe the four key components dictating the germination of seeds, and gain an understanding of how young seedlings grow. So you may already be familiar with the crop life cycle in its most basic form, but just to kind of go over it again. So a lot of horticultural crops develop from seeds. There are notable exceptions, and we will go over those in future screencasts. But yeah, the most basic crop cycles begin with the planting of the seed. This then germinates to give rise to a juvenile plant or seedling. The plant then grows to reach adulthood, and in some cases this will be the point at which the fruits are harvested, for example. And some crops are harvested actually before they reach full maturity, and again we'll go over some examples of those. Then after the adult stage of the plant's life cycle, following flowering, the plant starts to enter its final phases of life, where it then begins to senesce. So this is the process by which the fruiting bodies of the plant begin to mature and drop off, and the seeds are set within fruits or pods, and growth generally ceases at this point. This is then followed by the death of the plant, and the following generation is then continued through those seeds which are produced through the adult and senescence phases of the crop life cycle. So when we're talking about horticultural crops and seeds of horticultural crops, there's a huge diversity of different types and shapes and colours of seeds. So obviously this reflects the diversity of horticultural produce, but all seeds contain the embryo of a plant, and depending on the sort of evolutionary history of that particular crop species, the size and shape of the plant has evolved or been modified by human breeding efforts to withstand certain unfavourable conditions. And seeds can remain in a state of dormancy for many years in a lot of cases, and it's even been possible to revive plant seeds after many hundreds or even thousands of years. Growth is usually triggered when environmental conditions are generally favourable, and there are internal mechanisms within the seed that prevents them from germinating at inopportune or inappropriate times of the year, for example. So seeds have a variability in terms of what's called their viability, so their ability to germinate. So viability is the potential for germination. A seed can be described as quiescent, where the seed is not viable and it does not germinate because it's not the correct conditions for its growth have not been met. Dormant seeds are have a slightly different definition. So the seed does not germinate until physical or chemical barriers to germination are overcome, and these are sometimes, for example, a thick seed coat needs to be broken down over time, or certain chemical changes need to take place within the seed in order for that germination to happen. And in this slide we just see lists of some horticultural crops varying in their the length of their average sort of seed viability. So we can see down at the low end, with 1 year of seed viability after harvest, we have crops such as onion and parsnip. So these generally need to be stored in appropriate conditions quite rigorously in order to preserve their viability. So this is quite important for seed companies, for example, who are selling onion and parsnip seeds. It's not really viable for them to store up those types of seeds for long periods of time, so they need to keep them viable for at least the amount of time it takes to sell seed for 1 year. Other crops, so the average kind of viability for any given horticultural crop would probably be about 3 to 4 years. So things like broccoli, spinach, carrot, chicory, pumpkin, just picking out some random ones here, but generally there's a lifetime of seed viability of about 3 to 4 years. And then you have much longer sort of viable seed types for different crops, so things like radishes and lettuce. So these need less care when it comes to storage. Nevertheless, most of these types of crops will be stored in quite rigorous seed storage conditions after they've been harvested. And those sort of conditions in commercial seed production, for example, generally require the seeds to be kept very cold and very dry. So this stops them from rotting, also stops them from germinating prematurely. And a good example of the conditions needed for seed storage are kind of represented by the Svalbard Seed Bank, which you may have heard of, which is a program for storing human food crops and the diversity that's contained within those. And it's located in Svalbard on an island that belongs to Norway. And there's a reason why it's on an Arctic island. It's because it's very cold. So that makes keeping the seeds under optimal conditions much easier. So in terms of home growth and gardening, for example, that same principle still applies. Packet seed is often preserved in a kind of foil packaging, which keeps the seed waterproof and airtight and ensures that the seed viability is maintained. So looking in more detail about the process of germination from the seed, there are 4 main requirements to trigger germination: water, oxygen, temperature, and to a lesser degree light as well. And we'll go through each of those in turn. So water is obviously a key requirement to germinating a seed. So in commercial productions, what usually happens is seeds are harvested from plants and they're dried down to about 10% moisture content. And this again helps preserve the seed and protect its viability. So what happens when the seed is planted, the water content of the seed has to be increased above a certain threshold. So it's generally above 70% to trigger germination in a seed. This absorption of water by the seed is called imbibing. So the water is imbibed by the seed and softens it up like a sponge, so it kind of absorbs the water and expands. And this triggers processes within the seed, which then promotes germination and growth. So this process can be taken advantage of commercially for growers to improve germination and also to improve the uniformity of growth. So, for example, this is a slide showing how artificial germination can be induced by imbibing the seed. This then in turn activates the seed, and activation period commences. And ordinarily that would then trigger growth, but during seed priming, the seeds are then dehydrated again, so they're dried back down to that 10% moisture content. And that dehydration kind of postpones growth and germination. And that then allows the seed to be stored again for a period of time. Then when it's sold to the growers, they sow it out into their fields. It's planted. The seed then takes up water again, imbibing the seed. And then that growth period starts pretty much straight away. So this is called seed priming. So the seed has been primed for growth, and it basically allows for germination times to be much reduced once the farmer has got the seed. So the second requirement for germination is oxygen. So the seed itself cannot photosynthesize. It has no chlorophyll, it has no leaves. So it uses the stored energy within the seed to grow. and this process is controlled by respiration, and obviously respiration requires oxygen. So seeds won't germinate in waterlogged soil, for example, if there's not enough oxygen present. Thirdly is temperature. So all species have a kind of inbuilt threshold for temperature sensitivity and what's considered optimum for any given species. And this facilitates the functioning of enzymes and respiration in the given environment in which that crop has evolved or has been bred to grow. So this generally ranges from 0 degrees centigrade all the way up to 40 degrees centigrade. So different species are adapted to different environments, obviously, and this goes back to their evolutionary origins, and domestication through plant breeding has adapted certain crops for certain other environments. But they typically only respond to a very specific temperature range. So, for example, this is a cucumber plant. Cucumbers generally require a minimum temperature of 10 degrees C in order to germinate. Whereas tomatoes, which come from South and Central America originally, have a much higher temperature threshold for germination, so that's 15 degrees C. At the other end of the scale, lettuce plants won't germinate above 30 degrees C, for example, so they're used to much cooler temperate conditions. And some cultivars will actually the seeds will become dormant at temperatures over 25 degrees C, so this prevents the seed from germinating at temperatures greater than 25. Some species have a very wide range which kind of encompasses the entire spectrum of temperatures for germination. So, for example, mustard seeds will germinate at temperatures anywhere from 0 to 40 degrees C, so long as the seeds are not allowed to dry out. Other examples, carrot has a germination range of between 10 and 30 degrees C. It's a temperate vegetable, so it can cope with the lower temperatures and can germinate quite happily at 10 degrees C. Whereas eggplant, which again is a kind of a tropical plant originally, it requires a germination range of about 15 to 30 degrees C. So light, while not strictly a requirement for germination, it can accelerate the germination process. So once the young cotyledons of the plant have sprouted, this allows the young seedling to begin photosynthesizing and creating its own sugars. So crops such as lettuce and celery generally germinate better in a partially lit environment compared to complete darkness, for example. This slide just shows that there are two main types of germination. So there's epigeal germination and hypogeal germination. So epigeal is an example of a crop which has epigeal germination, for example, is tomato. So this is characterized by the fact that the seed itself is carried above the surface of the soil as the plant grows. Whereas in hypogeal germination, the seed body remains below the soil surface, and the plumule containing the cotyledons then grows above the soil surface. So once the cotyledons have emerged, the seedling then begins to photosynthesize and grow. After the cotyledons have expanded, what's called the true leaves then start to emerge and start to grow. So this slide just shows here some tomato plants. The cotyledons are kind of oblong shaped. They don't have any lobing or serration on the leaves, whereas the true leaves, by contrast, have a much more well-defined shape and are typical of the leaf shape of the juvenile kind of stage of a plant's growth. And the plant will grow upwards according to the environmental stimuli it's exposed to. So the main cues are light, so this is called phototropism, and they grow away from gravity, so this is called gravitropism. So in the emerging seedling, the tropic response is controlled by plant hormones called auxins. So auxins accumulate on the shaded side of plants. So light breaks them down on the lit side and they accumulate on the shaded side. And that accumulation stimulates growth. So as you can see in this slide here, the auxin molecules accumulate on the shaded side and promote cell elongation on that shaded side. So this causes the tip of, or the meristem of the plant to start angling towards the light. And then this dictates its growth upwards and towards sunlight. And most plants, most crops have some form of this process to direct their growth. Roots and root tissues have the opposite response. So auxins inhibit the growth on the shaded side of roots so that they grow away from the light. So just in summary, most horticultural crops have similar life cycles. Seeds are the first life cycle stage and have a built-in mechanisms to control germination processes. Conditions must be favourable for growth, or the plant may die or may end up in a growth situation which is unfavourable for it. Different species have evolved mechanisms to ensure they germinate and emerge at the correct time, and this includes involving different temperature ranges for controlling germination. And young seedlings will utilise these environmental stimuli to grow and direct their growth.